everyone, today we'll be learning the effect of temperature on simple muscle twitch. Now, what is the significance of this experiment? Through this experiment, we'll be able to understand that the efficiency of skeletal muscle contraction will increase with the increase in surrounding temperature, but within physiological limits. Although the experiments were earlier performed in the labs, but now we'll stick to the theoretical aspects of the experiment because of the changes in the curriculum of MBS. What is the experimental setup? Talking about experimental setup, this circuit requires the drum-in circuit, which means the primary wires of the stimulator are connected to the knob of the kymograph. The secondary wires includes the stimulating electrodes, and they are utilized to stimulate the nerve muscle preparation. And the pointer from the nerve muscle preparation will be recording on the drum of the kymograph. Other setup remains the same as for recording of simple muscle twitch. The one change which is seen in this experiment is the utilization of Lucas chamber instead of myograph bore, because the nerve muscle preparation which is prepared is dipped in amphibian Rigor solution to see the effect of different temperatures on nerve muscle preparation. We also require a temperature recording instrument, that is a thermometer. Temperature which are used in our normal temperature, high warm temperature that is 38 degrees Celsius, and cold temperature that is somewhere around 18 to 20 degrees Celsius. We will be obtaining three curves at three different temperatures. But point to remember is that all three curves will be obtained from the same baseline, from the same point of stimulus, and from the same strength of stimulus. Now, we will observe the effect of warm Rigor solution before than that of the cold Rigor solution, because cold Rigor solution leads to inactivation of the muscle. Now, just looking onto the graph, we have got three different graphs. One showing the normal response at normal temperature, that is 26 degrees Celsius. Others showing temperature response at 38 degrees Celsius. And the third curve is depicting response at 16 degrees Celsius. We can clearly see from the graph, which is at 38 degrees Celsius, that by increasing the temperature of the Rigor solution, the latent period has decreased and the various phases of muscle contraction, that is the contraction phase and the relaxation phase, has also decreased. Now, if we look towards the amplitude of the curve, it is clearly visible that the amplitude of the curve at warm temperature is more as compared to that at the normal temperature and the cold temperature. From this graph, we can easily assess that by increasing the temperature around the nerve muscle, it leads to increase in the amplitude of the curve, which means the force of muscle contraction increases, and also the duration of various phases decreases, which means that the metabolic processes in the muscle occurs at a very fast rate. So, to record the effect of temperature on warm Rigor solution, we can say the amplitude of the curve will increase and duration of latent period, contraction phase and relaxation phase will decrease. Now, talking about the reasons why this thing is occurring. Warm temperature leads to increase in the conduction velocity of the nerve impulse transmission, and therefore this leads to decrease in the latent period. There is decrease in the synaptic delay. Further, it causes decrease in the viscosity of the muscle tissue. It accelerates the electrochemical processes in the muscle tissue, and it increases the metabolic processes in the muscle tissue. As a result of all this, the depolarization of the muscle occurs very fast, and after the depolarization, the muscle contraction response will be very fast. So we get a contraction phase for a short duration and followed by relaxation phase which is also short. So, latent period decreases because of increased velocity of nerve transmission and decrease in synaptic delay, as well as contraction phase and relaxation phases decreases because of increased depolarization rate of the muscle and also increase in the biochemical processes in the muscle contraction plus decrease in viscosity of the muscle contraction. All these things also leads to increase in the force of the muscle contraction, and as a result of which the amplitude is higher. By utilizing a cold ringer, all the responses which are seen in warm ringer will be reversed. So it is clear with this graph that the cold ringer effect will be seen in form of increase in the latent period and increase in duration of the contraction phase as well as relaxation phase and decrease in the amplitude of the curve. Now there is one important term which is known as heat rigor. Whenever the temperature of the muscle increases to more than 45 degrees Celsius, then at this certain temperature the muscle proteins get denatured. Actin and myosin filaments are unable to carry out their normal contraction phenomena and muscle becomes in a state of permanent contraction. This stage is known as heat rigor, and therefore while performing the experiment it is of utmost importance to note that the temperature does not rise beyond 40 degrees Celsius. So 40 degrees Celsius is the last maximum temperature which we can utilize for this experiment. But another term which is associated with rigor is rigor mortis. Rigor mortis is a permanent stiffening of the muscles which is seen after death. It is the third sign of death, third stage sign of death, and it occurs because of the depletion of the ATP. Without ATP, myosin molecules remain adhered to actin and thus the muscle fibers remain in a state of contraction. Thus, we came to know that increasing the temperature of the muscle improves the efficiency and performance of the muscle. How we can utilize this information in practical life? We can see the effect of warming up exercises. It is always advised to go for warm-up exercises before we are going for some strenuous physical activity or some sport or athletic event. And therefore, this warming up exercise helps to prime up the muscles for the physical activity by increasing the temperature of that local area of the muscle initially and of course finally the overall temperature of our body. Along with the increase in temperature, there is increase in blood flow because it primes our cardiovascular system also. This causes decrease in strain and decrease in risk of injury to the muscles. Increasing temperature by warming up exercises improves motor nerve transmission to the muscles, and also it increases the muscle metabolic processes. It decreases the viscosity of muscle elastic elements, and as a result of which muscle performs faster and in more efficient, and also the risk of tears and injuries is reduced. So this is the physiological significance of nerve muscle preparation, studying the effect of temperature on nerve muscle preparation. I hope the things are clear to you with this small video. I hope to see you in my next video. Thank you so much.